I am so super excited. This is my first Launchbox ladies event, and I'll tell you what, we have some fabulous ladies. I'm happy to be part of this team, but the women, I don't even want to read this but I will. And then I'll tell you what I really think. First, I'll tell you what it says. It says, first, I'd like to introduce you to Dr. Vassie Ware. Vassie's a professor in biological sciences at Lehigh University. Dr. Ware holds a BA degree in human biology from Brown University, a master's in philosophy, and a Ph.D. in biology from Yale University. And then she did a postdoctoral stint in molecular biology at Brown University. But before I ask you to welcome her, I want to say that not only are these women smart, brilliant, at the forefront of their careers, they're also really nice, kind, generous women who are nice to be with, and that is an added bonus. So please welcome Dr. Ware. And another lady, I feel like these are my new friends. I'd like you to meet Dr. Katie Worrilow. Katie is the founder and CEO of Life Air Systems, which we'll learn more about shortly. She received a doctorate in anatomy and cell physiology from the University of Virginia School of Medicine and completed her postdoctoral fellowship in reproductive physiology and infertility at the University of Pennsylvania School of Medicine. Please give Dr. Katie Worrilow a warm welcome. Okay, again, I'm absolutely thrilled to have both of you here. And so maybe we could start by asking both Dr. Ware and Dr. Worrilow to talk a little bit about their background. Where did that come from? I don't know. I'm tongue-tied today. About their background. Forgive me. I don't know. I don't know where that came from. Sorry. What are you going to do? What can you do? Okay, Dr. Ware, is that? I don't know. Really? That's the last time I'll be offered this position to MC. Next time we'll get somebody who...
Okay, well,
thank you, Dr. Ware.
Yes, I was going to start with the 20th century, but that was a better lead-in.
Okay.
I want to take you back a little bit of time. Just my mic. Okay. Is that better? All right. I'm taking you back in time. Perfect. To the 20th century. There are some of you, perhaps in this room, who weren't in the 20th century. But I want to go back to 10th grade for me, because that was when I had an epiphany in biology and life science. I had a biology teacher in the 10th grade, Mrs. Corbin, who presented biology in such a way that was unbelievably boring. And I wondered what she was looking at, because when I looked at life and all the unknown about life, I did not see anything that was stagnant. I saw all kinds of possibilities. And she just presented science, life science, in such a way that let us know, at least if you were asleep, that there was nothing to be learned. And I knew otherwise. And so, in a way, most of the time you hear people say, this person said this thing that made me go forward. She didn't say something that made me go forward. So that I'm starting you in the 10th grade. I then chose biology, not because I thought that was the only science that I could do. I was actually interested in sort of mechanical engineering in a way, because I like to take things apart, and I was always interested in how things could come back together and the mechanism by which things would work. And so I managed, though, to narrow my, my field of interest when I went to Brown, in thinking I would be in psychology, turns out, because I also had the opportunity in high school to resolve a number of conflicts between peers. And I think there were folks that thought, I used to think that I was wearing a sign that said, "Tell me," and then I could, of course, try to help to resolve the situations that were happening. So I started out in psychology thinking that this might be a field for me. And so I took a number of courses, physiological psychology, child development, etc. Loved it. And it was only when I got to personality that I knew psychology in that way was not for me, because anyone could develop a theory of personality. The person down at the corner of Molly's could have a theory of personality, and there was no way to test that. You know, you could not typically go in and modify brains and be able to ask if I could change someone's personality. And certainly we would want to do that with some of the figures that are out there these days. But nonetheless, that's when I knew that what attracted me to the life science piece of what I was studying in psychology was really the life science, the science piece, the things that were testable. So I became a biology major. And I had mentors at Brown that were really instrumental in getting me involved in research. I had an undergraduate research advisor. I was also a teaching assistant in a course that I had taken and had been quite successful in. And so I had mentors there that really made a difference for me in terms of steering me in a direction that I thought sounded great, right? And so Dr. Walter Quevedo, my Bio 1 teacher, was one that I really hooked on to when I was at Brown. And he encouraged me to go to graduate school, and I went to graduate school at Yale. And that was a really interesting experience in many ways. I joined a laboratory that was all male. It was quite large. The only female in the lab at the time was, the other female was actually the animal care technician. And we have a very funny story, which it actually almost has something to do with the bathroom. But yes, I will actually tell you just one little piece of this story, and that is that one evening, actually it was a morning, I had come into the laboratory to do some biochemistry related things, and I had a bottle, a huge 5-gallon type bottle of dimethyl sulfoxide, DMSO. And I was using that because part of the ability to see some of the proteins that I was working on required that I use DMSO and a carcinogen called PPO to make this solution to then do some imaging. Okay, I walked in the morning. It was pretty cold in the lab. I picked up the bottle. Thankfully, it was only DMSO, and the entire bottle just fell apart onto my lab coat. This is another reason why they tell you always have a lab coat on. But in the case of DMSO, it penetrates so quickly that I knew I had to disrobe as quickly as possible. And so I did. I disrobed, and fortunately, completely, fortunately, the person that walked into the lab that morning was Avis. And so, almost a bathroom story, right? Okay, at any rate, I got through the program at Yale in biology with a focus in cell biology, developmental biology, and genetics. Became very much interested in, how things work. That's always of interest to me. How does, okay, we can define all the pieces, but how do you put them together to make it work? And I kept thinking, Mrs. Corbin, this is why I knew you didn't know what you were talking about, because there's so much that we don't understand. Anyway, left Yale, joined the laboratory of my former mentor who was one of my instructors in that course that I talked about at Brown, in which I was a teaching assistant. So I went to her laboratory as a postdoc and learned some more about molecular biology and stayed there about 4 years, and then came to Lehigh. I've been at Lehigh since 1985, and I know that that surprises many of you when I say that, but that's been a long time since I've been at Lehigh, and all kinds of things have happened in my career at Lehigh. The research piece still fascinates me. I mean, I would give anything to be able to spend my entire day in the laboratory, but then I would miss out on all the student interactions that I have in the classroom, which I love. And then I would miss out on all the administrative things that I do that mostly I love. But I have to tell you that the life that I have been able to unfold, you know, since the 20th century into this century has really been stimulating for me. And I can't actually thank many of you enough for giving me an opportunity to share some of this with you. So I'm going to stop there.
Can you hear? Oh, okay. I'll put it on mute. Well, first, thank you very much. It was fascinating. I'm looking forward to more conversations and our mutual love of biology. And thank you to Launchbox Lehigh and Launchbox ladies and each of you for your kind invitation to me to join Dr. Ware and each of you today. Looking forward to a great and lively discussion about bathrooms. So, I feel a little ill-prepared. I'll make it up as I go along. But my path to where I am today, it was anything but a straight line. And there were, as a scientist, if I picture a graph, and I think of the ups and downs and twists and turns, and what's been amazing is just learning from those twists and turns and ups and downs of that line to where I am today, and just learning from that and help to use that information and data for your next positive step. So my genesis, I'm a reproductive physiologist by training, and it has been really an honor and a privilege to serve as a clinical provider of human in vitro fertilization, or IVF, and to work with amazing individuals and couples to help them fulfill their dreams of having a family. And I did that, served that role for well over 20 years, and loved it, absolutely loved it. We would do anything for our patients. And as part of that role, let me think, 18 years ago, I was asked to design and build a new IVF laboratory and program for a major hospital. And in doing so, we thought, well, you could build kind of your traditional IVF laboratory, your standard laboratory space, or then we assumed that air quality had an impact on the growth of the human embryo. I mean, you're culturing them outside the body for 7 days, trying to mimic the in vivo environment, which is extraordinarily difficult to do, especially with a cell that sensitive. And so we proposed culturing our human embryos in an ISO 5 clean room, thinking that would remove the variable of air from our process. And so to their credit, because it was far more expensive, the hospital said yes. So we actually designed and built the first ISO 5 clean room from the ground up in which to perform human IVF in the world. And we started out of the gate with clinical pregnancy rates over 20 percentage points higher than the national average at that time. And that data represented about 40 to 60 couples. So we thought, this is unbelievable. We thought everyone should do IVF in a clean room. And so we were thrilled. And then that pattern repeated itself in a second quarter. So now we had data on about 80 couples, 100 couples.
And then in the third testing quarter, we dropped from about a 51% clinical pregnancy rate to about an 8. And in our mind, nothing had changed. So imagine you've counseled a couple 6 months prior, and you've told them that your success rate is over 50%, and now you're at 8. And the worst, when they're cycling. And the worst part is you don't know why. And we did everything any good IVF program would do. We looked at our cell culture media, our protein, patient etiology, our own skill sets, all of our SOPs. We ripped everything apart. But we didn't look at the air because we were in a clean room. So fast forward 10 years, and there was this pattern continued. We'd have great success, and then we'd have tremendous failure. Great success and tremendous failure. And this was personally and professionally completely unacceptable. Because I don't know if any of you are familiar with IVF, but it is an extraordinarily difficult process to go through in every way. And for our patients to have these dips, and we weren't alone in this in the world, but for our patients to have these dips was, without our understanding of what was causing it, was beyond unacceptable to each of us. So there was one evening I was leaving the hospital, and they were resurfacing the medevac pad. As a level 1 trauma center, they had 8 medevacs, and they were resurfacing the medevac pad. It's like 6:30 at night. And I noticed the fumes, and we were in a dip again. So I noticed the fumes coming up from the medevac pad, and I stopped, and I just said, "When did you start this process?" And they looked at me like they really did not want to talk to me. They just said, "Just please go home." But they told me, and then I said, "Do you have an MSDS sheet on this asphalt?" And they said, could you just call tomorrow morning? You know, we just want to get the job done. So I did, got that sheet, and then we learned what airborne chemical pathogen was being produced by the asphalt, toluene. So then we went back, and because we were a clean room, we were collecting live airborne pathogen data, only because we wanted to be certified as a clean room. So we then went back and looked, and we had parts per billion levels of toluene in our clean room space, coincident with the asphalt, coincident with our drop. That made us look back at all the other 10 years of data, and we saw this same pattern repeating. So despite being in a clean room, and despite everything that we'd poured our hearts and soul into and providing the best environment for the embryos, we had this breach. But you don't know what you don't know. And so once we learned that, I went to the CEO of the hospital and said, "I think we have figured it out. The human embryo, or frankly any living cell, needs airborne metrics beyond what a clean room offers. A clean room doesn't address your chemical airborne pathogens, and it doesn't address A, B, C, D. Now we know we need that." And he said, "Great." And he actually gave me a blank check, only one in my entire life. And I couldn't use it. It was so sad. I thought it'd be the easiest thing I ever did, go shopping. And there was nothing commercially available that I could purchase or that we could purchase as a practice to deliver the air we now knew the human embryo needed. And it was really the absence of a solution that led to Life Air Systems, because it was, again, unacceptable. We had identified a problem, the significance of the problem, and there wasn't a solution. So that was completely unacceptable for our patients. And that's really what led to the genesis of Life Air. It was completely unexpected. Like I said, it was really just a path of problem solving, and we would do anything for our patients, and that remains to this day. So.
Katie, did you want us to show your video?
Is there time?
It's queued up.
You sure?
Yeah.
Okay.
Mhm. So we're going to watch a brief video, and then I'm sure you all have lots of questions.
A new breakthrough is leading to dramatically higher pregnancy rates through in vitro fertilization. The good news for millions of Americans facing infertility is the result of a new technology from Life Air Systems. For years, unexplained periods of decreases, baffling researchers. LifeAir Systems founder, Dr. Kathryn Worrilow, a leading embryologist, spent 2 decades researching the causes.
The problem, the challenge, is not knowing the cause of such a decline in your outcomes. In IVF, there are countless variables that can impact successful outcomes, but one that we couldn't even see would prove to be a variable extraordinarily difficult to control. The variable? Air. There's construction outside, or a fire 2 blocks down, or they've resurfaced the parking lot at the hospital or a restaurant nearby. Not to be able to say to those patients, no matter what happens outside on the day you arrive for your egg retrieval, your embryos will be protected, and despite operating within a clean room, we still couldn't say that. That, to me, is completely unacceptable. IVF is an extraordinarily difficult process for the couple, emotionally, financially, medically.
Worrillow's team of physicists, chemists, and engineers developed the new smart technology that systematically remediates all biological and chemical pathogens. Where other purification systems filter down to parts per million, LifeAir reduces to undetectable parts per billion. It's so powerful, it neutralizes anthrax. The result? In an industry where a single percentage point is significant, IVF programs across the U.S. using the LifeAir technology are experiencing double-digit increases in positive outcomes, pregnancy, and birth.
They told me about the LifeAir system, and we just thought it was wonderful because you put so much, into this process and the cycle, and if anything's going to help, your chances of achieving pregnancy, we wanted it.
We were looking to build a new center, and we knew that the air system in the center and the air quality was crucial to both the health of the embryo and ultimately the patient's success. I realized that the LifeAir system truly would be the absolute best outcome that we could offer to our patients.
When we were discussing about the LifeAir, it kind of made me feel a lot at ease about how successful we would be.
Unbelievable, the difference in the pregnancy rates. And the quality of our embryos, fantastic.
Providers are hailing LifeAir systems as a new tomorrow for those facing infertility. For LifeAir Systems, I'm Victoria Hicks. A new breakthrough is leading to dramatically higher pregnancy rates through in vitro fertilization. The good news for millions of Americans facing infertility.
Such incredible stories, and not quite where you started, not really where you expected. Vassie, when we spoke, you told me that 2 of your research interests were hobbies. Can you talk to our group about that?
Okay, sure. In fact, just one is a hobby. I'll tell you a little bit about how this transpired. As a graduate student, and this is very funny because I was in a laboratory, a reproductive physiology laboratory, as a graduate student, yes, at Yale in Clem Larkert's lab, who worked on, chimeric mice. Okay, this is true. We're not making this up, okay? At any rate, I became very much interested in what genes would be expressed really in development. So if you think about a single fertilized egg, a single cell, and we know what happens. There are divisions and all kinds of incredible things that happen to give rise to a multicellular organism, as we all are. But I was very much interested in the genes that get turned on to allow this to happen. And I'm still fascinated that in most instances it actually works out. So as a graduate student, I was always interested in, again, gene expression, the regulation even of gene expression. What times in development do certain genes get turned on, and what happens, and what's controlling all of that? As a postdoc, I became interested in, again, the molecular aspects of that. What are the mechanisms by which these kinds of things occur? And so my laboratory at Lehigh really focuses on a component of the gene expression pathway. It's called the ribosome, and for maybe some of you have heard of a ribosome, and it's really a machine. It's an amazing machine in cells, and people tend to think of this machine as, it's responsible for synthesizing proteins, and they sometimes can ignore it as, the ribosome, it synthesizes proteins. But it turns out that ribosomes are not all alike, and this is something that is coming out, a very new development in the gene expression field, that you need to think of it as like the models of cars. We know that cars, there are lots of, I'm looking at them now out the window, lots of different car models. They all get us from point A to point B, maybe not as smoothly as all of them, but nonetheless, we can identify them as cars, but they don't look alike, right? They don't all look alike, and that's the same thing with ribosomes, which we're beginning to understand. Now, I'm not going to go into the details of that because that's actually the work that I primarily do, having started this as a graduate student, going into my postdoc, and now developing that as a primary research interest at Lehigh. However, the hobby situation is this. About a decade ago, I applied for a course grant from the Howard Hughes Medical Institute. And for those of you who have heard of Howard Hughes, very unusual character, I will say. And I can't even imagine that Howard Hughes imagined the impact that the funds, the monies that he accumulated would have had on society at this time. It's phenomenal what HHMI does to support biomedical research and science education across the world at this point. But I applied for a grant, this course grant from Howard Hughes, to have Lehigh participate in a program that's called C-Phages. And the goal of this program, believe it or not, is to get first-year students involved in research. And one of the reasons that this is important is because it's been shown that if you can get a student hooked early in research, the odds of retaining them in STEM really go way up, way up. It's like high air quality, right? High air quality here. Get the student engaged in research. They'll maybe stick it out when they are in some of these gateway courses that make you want to pull your hair out. All of that, the persistence piece is there, okay? So I wanted Lehigh to participate in that. So our students had that opportunity. And the way the program works is that students go out into the environment and they get a soil sample. You know, I know someone's grandmother here said, don't eat that dirt. But it's because there are all kinds of things in dirt, including anthrax. But nonetheless, there are all kinds of communities of organisms there. And there are viruses that infect bacteria that are in the soil. And so the students bring in a soil sample. They then extract from the soil viruses that infect any number of bacterial strains that we can use. The strains that we started using were related to Mycobacterium tuberculosis. I know everyone's heard of that, tuberculosis. Our students don't work with Mycobacterium tuberculosis. They, in fact, work with Mycobacterium smegmatis, a cousin, okay? And so the students then have an opportunity to isolate these viruses. They get to name the virus. They get to extract the DNA. They look at the electron microscope, through the electron microscope, to see what the form of the virus is like. Those genomes get sequenced. They then learn bioinformatics and computational tools to then find the genes in those particular genomes, and then we can learn all kinds of things about these viruses. Now, we tell the students a decade ago when this program started, you just never know where this information is going to go. The viruses that you isolate, some of them can attack and kill Mycobacterium tuberculosis. You tell the student that, and it's true. It is actually true that some of the viruses that they can isolate have that capacity. But you never really know if it's going to go forward in time to really have that kind of impact. I will tell you that now, because of all of the antibiotic resistance that everyone hears about, and this is really a very serious problem, and I hope people don't ignore it, because antibiotic resistant bugs could be our big meteor that kind of wiped out the dinosaurs. If we have so many antibiotic resistant bugs that can't be killed by antibiotics, what will we do? Well, phage, viruses that infect bacteria, are really now becoming the conversation piece, phage therapy. And so our students get involved in this program because they have in the backs of their heads that phage therapy really is going to save us. And that's probably true, because out there in the wild, we probably have a greater probability of collecting viruses that are going to infect bacteria than we do in producing yet finding other antibiotics that are going to work. So those two processes of trying to find new antibiotics, working with phage therapy, are probably what's going to happen. Now, I'm telling you all this because, remember, this started as a course, right? And so over the years now, you know, I was thinking, this is kind of neat. I like this. Phages are interesting because they have some amazing gene expression mechanisms, and I'm interested in gene expression. I'm interested in gene regulation. I can do this, right? It's a hobby for me, I thought, until the questions that come up become so amazing. And I had a student, Joseph Tiem, in fact, was one of my students in the phage program. And one day he asked a question. In fact, I teach this phage program on Tuesdays and Thursdays. And right before I came here today, I had a group of students that I was talking about Joseph. And I wanted them to appreciate that their questions can really take you amazing places. So this young man, some years ago, asked the following question of my colleague and me in class. We were talking about the ways in which viruses kill bacteria. And I won't go through that lecture for you, but the student asked, was it possible that another type of virus could infect a particular type of bacteria that we had in the laboratory? And it's a little bit more complicated than I have just said. But my colleague and I said, we don't know. Now, the student was, in fact, horrified that we said, we don't know. But I could also follow up by saying, no one's done that experiment. That's why we don't know. No one's done the experiment, but you can do the experiment. And that's what happened. He did the experiment, and then, ta-da! What do you think happened? A whole new field of understanding what happens when viruses that infect bacteria that don't kill them immediately, they hang out in the cell, okay? Imagine hanging out in the cell. And you now, as a virus that's in this cell, have the opportunity to protect that cell from being invaded by other viruses. Is that awesome? Is that incredible? So there is viral defense against other viruses. My student asked a question that allowed us to understand that. This is why my hobby now became a real interest, and we're pursuing those kinds of things because it really makes a big difference in uncovering all of these new models of understanding how these things work. And so you understand why I can't even imagine not being in the lab doing the kinds of things that we do? But that's how I got to have the hobby sort of transform some of the work that we do to become very practical in nature.
Thank you. You can hear the passion in both of your voices, the excitement about what you do. Katie, do you want to talk about the overcoming failure and how your career path twisted? You told us a little bit, but do you want to talk more about that?
Sure. Actually, I'd like to talk about viruses, too. Your class is Tuesday and Thursday. I might want to sign up for those. Yeah. Oh, my gosh. No, there are a million things I want to talk to you about. Yeah. Okay, so failure. Not bathrooms, failure. So.
No, you're not. It's okay. It's good.
So if you recall, at the end of the 10-year period of the clean room, we were just understanding the metrics of air that we needed. And I was given a blank check, went out to purchase something, and right at that same timing, and this is it's a continued wave in human IVF, groups are forming large corporate groups. Individual practices are forming large corporate groups. And we had no idea. We were just focused in the lab, working with our patients ourselves. And our practice was purchased by a large corporate group. And just in the blink of an eye. And it was just carved out of the hospital. So this was extraordinarily difficult because we had poured our hearts and souls into this environment. And now, I mean, the environment had taught us so much about what the living cell truly needs to thrive and be successful. And so the practice was completely uprooted, removed from the hospital, sold. A few in the practice went on with the new group. But I went to my national meeting in October, it's every year in October, and typically we were presenting negative data from our clean room, and negative data can be phenomenal, because negative data can teach you a great deal. I don't like that word negative, but our cellular failures in the lab is what, led us to where we are today. So my colleagues at the conference said, so what are you going to do? Because, they knew this was our heart and soul in this clean room environment and all the learnings. And I said, I'm honestly not sure. And they said, well, we'll give you a suggestion. They said, every year you've presented negative data. If this airborne pathogen, then we have this consequence in the human embryo or in our clinical outcomes. And they said, we all had the same issue. Why don't you develop a solution? And it was really the loss of our laboratory and the loss of our practice, which was devastating. I'm not going to mince words. It was devastating to all of us. But that loss led to, kind of a reevaluation of everything, suggestions of colleagues, using that data, using the pearls from the laboratory to come up with a solution. And again, a straight line, not having a straight line is fine because you can learn from that and keep moving forward and make positive decisions for your next step. One thing that's happened because we were very selfish in our goals with this air purification system, we were completely focused on protecting the human embryo. So we set the bar very high, and that's why we chose the anthrax spore to kill. Because if you can kill anthrax, you can kill C. diff and MRSA and TB, influenza, all the things that really concern us other than anthrax. And we also set the bar high in delivering below detection airborne chemical pathogens or your volatile organic compounds. If you can smell your lunch right now, that's parts per million, but it's parts per billion that the living cell needs. So what's happened is, obviously our genesis is in vitro fertilization, where we have our systems throughout the U.S., and we have 2 in China, 1 in Canada, and our first just went live in the U.K. But we're now, we have our first system in a hospital protecting their ICU from healthcare-acquired infections, killing C. diff and MRSA and influenza to keep those patients healthy, reduce length of stay, reduce cost to the hospital. We're also installing our first system in assisted living to improve their quality of life and reduce infections. And last week, and Alicia is here, we attended a very large biopharma conference called Interfex in New York City. And when you were talking about viruses, it is unbelievable. If you've heard of personalized medicine, cell and gene therapy, CAR T, which GoPen, they had a huge innovative hand in. It's unbelievable what they're doing in taking individual cells from your tumor and then developing viral vectors that then dissect the genes. They merge your patient's cells with the host cells, then re-inject them back into you for a personalized vaccine, if you will. And they're targeting specific cancers, hemophilia. I mean, there are a number of diseases. This is only the beginning. And because they're having to culture live cells in a very similar to way that we did with human embryos, we were meeting with a number of them in sharing with them our learnings from cellular failure and what they could do to improve their outcomes so that they can deliver a solution to their patient. So you can never learn enough. It's absolutely fascinating. And the questions that you ask direct the next path. And there will never be a time where there aren't questions to be asked. I mean, I'm still to this day fascinated by the black box of human physiology. There's so little that we know. We think we know a lot, but we don't. And just my fascination was molecular crosstalk between the sperm and the egg. And again, just keep asking questions and follow what you love.
Thank you. How about that for advice? And it's a perfect segue, Katie, to allowing the audience to ask some questions. So before we let these ladies go, does anyone have questions for them? I'm sure you have lots.
I can talk loud.
Can you?
Yeah,
Well, I want everybody to hear you. Thank you.
Sure. First, thank you very much, foremost. I'm going to start my question. You're going to laugh, and then you'll hear the question. I have a 15-year-old daughter. She is in love with the sciences right now. She thinks she knows what she wants to do, how she wants to do it. With things changing so much, not just in the traditional sense of education, but of course what is out there. What is the best advice for a parent of a young woman, desiring for the sciences, and how I can help encourage her as she makes that next step to decide? She thinks she wants to be a dentist right now. Maybe so, maybe not.
I can't wait to tell you a dentist story.
Oh, excellent, excellent. But how can I do my best to encourage her on the next right steps and paths?
Might I speak about the dentist thing? Yes. So many years ago, in fact, it probably was the 20th century, a student of mine at Lehigh she came and said, I'd like to get a BS in molecular biology. She was in the 7-year dental program with Penn. I want to get a BS in molecular biology, but I want to do it in 3 years. And I'd like to do research. And I'm hearing all of this as an advisor, and I said, well, Trish, in order to do the BS in 3 years, you will have to overload every semester, and that's possible. But be prepared that you'll have to take more courses than what is normal. I said, the research part, though, this is going to be a challenge for you to be able to take all the courses, the required courses, but yet have an opportunity to do the research. And I asked her, why did she think she wanted to do research? And this had to do with, this epiphany, it wasn't really an epiphany for me. I thought about medical school for 10 minutes, sophomore year, 10 minutes. And I knew that wasn't for me. It had nothing to do with the rigor or whatever. It had to do with the creativity piece, understanding I like it when I can discover as opposed to perhaps the practitioner part of it, right? When you need a hip replacement, you do want the person that's done 1,000 hip replacements, not the one that thinks, what does that look like, right? Okay, so anyway, getting back to Trish, I said, Trish, let's go forward. Let's try to see if we can't get all that you're interested in this package. And what I suspected was going to happen was that she was going to abandon dentistry because she would get engaged in a research project and be overwhelmed with it, which is what happened. So she did get her bachelor's degree in molecular biology. She went to graduate school at Yale in the molecular biophysics and biochemistry program, got her Ph.D., went to University of Chicago, did an outstanding postdoc there, got hired at Brown as an assistant professor, went to Arizona to lead the Department of Biology at Arizona, and is now the Dean of Natural Sciences at the University of Massachusetts in Amherst. Our running joke is that I saved her from being a dentist. And the reason I'm telling you the story is because what students will discover is that they can do a lot of things, and there's not one narrow thing anymore. There's so much multidisciplinary work that happens. And so I think the best advice is almost no advice, to try not to steer them in one direction, because things change so much, and what you will see, the interest that she has, the questions that she has that are burning, I think those are great. And you can encourage that. That's the kind of thing to encourage. But try, I think, not to limit in terms of what we understand right now as a career. Because when we started, the independent scientist in the laboratory was sort of the thing, right? We don't solve big problems anymore as an independent scientist. We have collaborations. We have all kinds of people at the table that have different expertise. We have to have the computational people. We have to have the molecular biologists. We have to have the engineers that are doing some applied aspects. So what I think will happen is that she will see the interest that she has will blossom in ways that will surprise her, as opposed to thinking narrowly about how she can apply it right now. Just let her keep going with all the questions, and I think she'll open up to, wow, I didn't know I could do that. That's what I think will happen.
Katie, you had recommended some recommendations for career advice in terms of business courses. Do you want to tell them that?
And I 1,000% agree with what she said, absolutely, because you can't predict. And there are so many opportunities. And the main thing is just building that foundation. And I studied what I loved. I wasn't exactly sure. I was thinking, med school or graduate school, but I wasn't sure which path. But, just math, science, I just took what I loved and just build that foundation. One thing I did not take was, because I was very math, science, engineering, so I was very heavy in labs, so there's not a lot of time. But I wish I had taken, and I tell everyone this, any kind of introductory, even if you think science is my world, which it was for me, take a basic business course, take econ, take any introductory level, whatever you can, wherever you are. I didn't, just lack of time, and I thought, my world was going to be science or healthcare or, research. So there are many days for me, it's like drinking from a fire hose. But I have an amazing advisory board and board of directors, and all, very well knowledged in the School of Econ and Business. And so I listen and learn, but that would be advice just to help round things out, even if she thinks she knows where she wants to be. Just keep the doors open and build the foundation.
Thank you. Any other questions out there?
Yeah, sure. Hi, I'm Tracy Damiani. You ladies are amazing. Oh, I'm loud. You ladies are amazing. Launchbox, you've taught us today, and I hope everybody remembers, is thinking out of the box, right? and what you said today is true. And just a question on LifeAir, are they using this in technology like Silicon Valley, NASA? Are they using it, they're trying to clone mammoths to keep the permafrost down and create new savannas in Russia so that the permafrost is more of a threat with carbon in the atmosphere than actually any of the industry we have? Are they using it in things like that?
Well, our technology is still relatively new, and so our launch was in human IVF, and we're just now in healthcare and soon long-term care, and then the personalized medicine. So we haven't. Absolutely, absolutely.
Thank you. Any other questions? Yes.
Speaking to us. So right now I'm trying to pick between two paths.
I'm sorry.
Thank you.
Thank you.
So I'm very interested in viruses and bacterial pathogenesis, and I probably talk about it every chance that I can. But I also love molecular biology, and so there's two different paths that I can take. I can do immunology and infectious diseases, or I can do biochemistry and molecular biology. But I want to do both. And so it'll take me longer than 4 years to do both. And people keep telling me that it's harder to get into respectable graduate programs if you take longer than 4 years to finish your science degree. So I don't think I can double major in both. And I just have so many interests that I don't know what to do. And I don't know how to narrow it down and pick a path that'll let me do both. So do you have any advice for that?
All right, I have a bias. I do have a bias. And it has, so the immunology piece of this, to be honest, at the undergraduate level, to get really a deep dive in immunology is exceptionally rare. I will just say that. It's rare. What parts of what you talk about that are more foundational are the molecular biology, biochemistry piece. You could almost go anywhere in a life science program with a good foundation in those aspects, you know, cell biology, molecular biology, biochemistry. That gives you something to work with. I almost want to say that the immunology piece, you learn aspects of immunology in molecular biology, cell biology, that would be, in most cases, sufficient to get into a graduate program in immunology. So you don't necessarily have to major in immunology as an undergrad. You would get enough foundation in those areas to really be able to be competitive for an immunology program. You know, I realize as I'm talking to you, I just have so many stories, I can't get over it. I have an immunology story, you know? No, but I'm actually thinking about years ago having taught an introductory cell molecular biology course, and I was team teaching with a faculty member. I had to do all the cell molecular part, or most of the molecular parts, and he did most of the cell parts, okay? And I live in New Jersey, and I had driven to Lehigh this particular day in my craziness, my crazy life, got out of the car and realized that I had no notes, okay? I had no notes, no slides, no nothing. And I had to do immunology that day, cellular immunology, all the cytokines, all of the, and I had to do all the rearrangements in the molecular, you know, what happens in that vein. And I thought, my first panic, and then I thought, wait a minute, you know this. And I just went in, and instead of having slides and all of that, I just had, you know, I wrote on the board, and I did it. So my point is, this was in a cell molecular biology course where I was delivering the basics of cellular immunology and the molecular aspects of immunology, which you would get in that kind of preparation. So I really think don't panic, all right? Don't panic. You will be fine if you, I think, get the good foundation, the understanding of the molecular, the biochem pieces of those. That's going to be more than enough to get you into an excellent graduate program.
Can I add something? Like another piece. So this summer I'm supposed to go to Yale to actually study more biomedical research, but I think I'm going to be paired in viral and bacterial pathogenesis. So would I still do things over the summer related to that, even if I study biochemistry? Yes. Okay.
Yeah, I mean, the point is, you know, we try to put labels, we put labels on, thank you. So the viral pathogenesis, you know, host-pathogen interactions, you could maybe be studying the cellular aspects of that, or you could be looking at some biochemical components related to that, you know, molecular components related to that. So it's very much multidisciplinary when you're thinking about it. When you think about a viral pathogen interaction and you really want to understand it, you absolutely have all those pieces, the molecular part, the cellular part, the biochem. I don't know what they'll focus on, but you're not going to be hurting by pursuing that as a research thing. And I that's so exciting to me. When you come back, come over to Lehigh and we'll talk too, okay?
Yeah, wonderful. I think we might have time for one more question. Is somebody?
Oh, I was just going to add.
you're gonna add? I'm sorry. Thank you,
No,
Katie.
no, that's all right. I completely agree. And also, for that foundation and what she was saying, but also once you're in graduate school, I mean, the first 2 years are pretty much defined, whichever program you're in. But after that, while you start your doctoral research, you have the opportunity to take a number of specialized and more advanced courses. So you, if immunology is still a love, you'll have that opportunity. But the main thing is to get that molecular cell biology and biochemistry background, and then that'll set your foundation.
Thank you. Thank you. No more questions for right now? Okay, Dr. Richardson, would you like to join us at the podium?
I just wanted to say thank you very much for a very dynamic discussion. I have the good fortune of having you both as friends, and I too have stories, but I will refrain from sharing those stories from Lehigh.
Together.
Yes, we have stories together. And so I just want to say that we promised this group a discussion about science, healthcare, and innovation. And I think the two of you epitomized that conversation very well. And thank you very much. The last thing that I wanted to say is I believe there may be alum from Brown in the room. Are there any alum from Brown? And are there any alum from the University of Pennsylvania? All right. So I just want you to know that I think you did your institutions fine. So lastly, the Medical Alliance. Are there any Medical Alliance members in the room? And we want to thank you for coming out as well. This was a great discussion, and I hope you can stick around for a few more minutes if people have additional questions. Okay.